Welcome to a new episode of Science, Technology, and Society Talks. For today's episode, we will talk about vaccines. Joining us here today, we have Cheina, Jaime, Archie, and Dane to discuss the following topics: the history of vaccines, how vaccines are created in the past versus today, role or influences of science and technology in creating the vaccines, and how vaccines affect our lives today in general. Without further ado, let's jump in right into the video.
Smallpox is one of the deadliest diseases in history. Not only did it claim the lives of over 300 million people, it even resulted in the decline of the Aztec and Inca empires. Luckily for us, the discovery of the smallpox vaccine and global efforts in immunizing people against smallpox led to the eradication of this disease in 1980. Before the creation of vaccines, people already had the idea of protecting themselves from diseases through intentional exposure. This practice is called variolation. Variolation is a method of inoculation that was practiced during the 15th century as a way of protection from smallpox. Through this method, dried smallpox scabs were blown up the nose of the patient for infection. Even though this was effective for some people, it's still a very risky method because it could lead to the death of the patient and spread the disease even more. The smallpox vaccine was the world's first successful vaccine created by Edward Jenner in 1796. He inoculated James Phipps with matter from a cowpox sore. Because of this, Phipps became the first human to be immune to smallpox. This method of inoculation is what we know now as vaccination. Vaccination was seen to be a much safer method compared to variolation because, in this case, cowpox had the same immunities as smallpox, but it was milder, making it less deadly to humans. Since then, vaccines have been created for many kinds of diseases such as polio, chickenpox, measles, and many more.
Hi, it's me. I'm Jaime, that's me. So, based on the previous presentation, you might ask, are vaccines conventionally safe or are they eventually going to turn us into zombies? So, we've all been there, we've all been concerned, but the first one is a much more valid concern than the second one because the second one is, I might say, an active imagination in our part. So let's break it down, shall we? So what makes the vaccine from before different from the vaccine now? So conventional vaccines are made using viruses and can take years to successfully be created due to the process of collecting the actual viruses and adapting them into the lab. Each new conventional vaccine back then requires a custom production process, and it includes complex purification and testing. However, right now, other vaccines use genetic code rather than the virus itself, with a technique using the messenger RNA. So what does this do? So the messenger RNA is basically it creates a genetic recipe and uploads the template in the lab so that it can be created from all over the world. So what the genetic recipe does is that it teaches the cell how to react when the pathogen enters the body, as opposed to conventional virus vaccines in which they're inoculated small amounts of viruses until the body learn how to be immune when the virus is reintroduced into their body. It is important to note that the vaccine development have come so far, and although there has been an issue like the dengue vaccine, that doesn't mean that vaccines are ineffective in itself. So when an anti-vaxxer tells to you that it embeds you with microchips and it turns you into a zombie, don't believe them. Just search the internet and it will reveal to you more than conspiracy theories in Facebook could reveal. So, why do we say that vaccines are conventionally safe right now? Because the path researchers must take with the potential vaccines is clearly defined and overseen by the Food and Drug Administration of different countries. Once researchers create the potential vaccine, the producer must apply it to the Food and Drug Administration, describing the product, the manufacturing process, and its effectiveness in the animal testing. So once the animal testing is done, the vaccine can begin 3 clinical trial phase. So the first one being evaluating the vaccine's safety and ability to generate antibodies in a small group of people. And we proceed to phase 2, which involves possibly hundreds of people to determine the right dosage levels that need to be given to effectively immunize the person from the virus. And the last phase will be inoculating it to thousands of people to analyze the safety and effectiveness of the drug. So before it is actually released to the public, it must undergo a series of reviews and regulatory approvals for efficacy, safety, and manufacturing. So back to the questions. Are vaccines conventionally safe? The answer is yes, because even when back then when the vaccination is much more riskier, it helped eradicate a lot of diseases which are considered extinct right now, and we should be grateful because at least our babies right now can feel the safety because there are no polios and measles that can bother them because they're already vaccinated, a privilege in which some of the previous generations didn't enjoy. So, if you're not vaccinated yet, try getting one and you'll learn more about vaccinations. So in the later part of the video, so stay tuned.
Hello, I am Arki, and let's talk about the roles and influence of science and technology in creating the vaccines that we all know today. To better understand the technology used for creating vaccines, let's identify first the different types of vaccines according to the US Department of Health and Human Services, and discuss the origin of some of these vaccines. So the type of vaccines according to HSS are the following: inactivated vaccines, live attenuated vaccines, messenger RNA vaccines, subunit recombinant polysaccharide and conjugate vaccines, toxoid vaccines, and viral vector vaccines. In the 19th century, Salmon and Smith pioneered the inactivation of an organism and the injection of immunogenic components. This is what we call an inactivated vaccine. Later on, Louis Pasteur first attempted to create the first forms of live attenuated vaccines, which were now adapted to modern vaccine technology by Enders et al. In the 1950s. When we say attenuated vaccines, it means that scientists use a weakened or attenuated form of the germ that causes the disease. So these vaccines are so similar to the natural infection that they help prevent. They are able to create a strong and long-lasting immune response. Examples of these vaccines used to protect against are measles, mumps, rubella, rotavirus, smallpox, and chickenpox, to name a few. So let's go forward to the 1970s. The ability to sequence DNA and the ability to express proteins in plasmids were the two significant breakthroughs that helped usher in the era of genetic engineering. So, what is genetic engineering? According to the National Human Genome Research Institute, genetic engineering, often known as genetic modification, is a technique that modifies an organism's DNA makeup using technology developed in laboratories. This could entail changing a single base pair, A to T or C to G, deleting a section of DNA, or incorporating a new segment of DNA. These methods were utilized to create the first recombinant vaccine, the hepatitis B vaccine, 10 years later in 1986. So recombinant technology makes it possible to generate the target antigen separately from the parent organism, eliminating the need to handle live infectious pathogens or potentially toxic byproducts of such agents. In effect, the quantity of antigen produced is increased. The vaccine's safety and the purity of the product are all improved. Efficacy is increased, costs are decreased, and potential side effects are reduced. As of lately, the effectiveness of numerous additional recombinant vaccines against diverse targets like malaria, hookworm, cytomegalovirus, parvovirus, and anthrax is currently being examined in clinical studies. Adjuvantation is the second major advance in the 1980s. So adjuvants are used to enhance an antigen's immunogenicity or to improve its presentation to the immune system. The mineral salts, calcium phosphate, and alum are the only adjuvants that have been given US approval for concurrent use with the vaccinations. 13 mineral salts are still used in some inactivated vaccines, but their efficacy is at best marginal. A highly efficient vaccine against H. influenzae type B, a major contributor to invasive infections in children including meningitis, was approved as a result of the application of conjugation as an adjuvantation technique. Another type of vaccine is the viral vector vaccines, which use a modified version of a different virus as a vector to deliver protection. Viral vector vaccines have been the subject of extensive research. Viral vector technology has been used in some vaccinations lately used to combat Ebola outbreaks, and several studies have concentrated on viral vector vaccines against other infectious diseases like Zika, flu, and HIV. This method was also employed by scientists to create COVID-19 vaccinations. So, all of these scientific discoveries and technological advancements have changed, improved, and added significant value to the production of vaccines. The aforementioned examples are just a few of many interesting influences of science and technology. Even for the future, according to the National Human Genome Research Institute, scientists are currently working on new types of vaccines. Two exciting examples are the DNA vaccine, which are easy and inexpensive to make, and they produce strong long-term immunity, and the recombinant vector vaccines or platform-based vaccines that act like a natural infection, so they're especially good at teaching the immune system how to fight germs. So you see, it's fascinating to know that as time goes by, our knowledge in health and wellness have been improved through extensive research and development. Science and technology continues to progress and find new ways in creating and producing vaccines for everyone in need.
Wow! The effects of vaccinations are substantial and far-reaching. The reduction of morbidity and mortality from diseases was formerly seen to be one of the major advantages of vaccination. And this perception still guides the development of new vaccines, especially in the events of outbreaks or against viruses that affects the most vulnerable members of society. Moreover, vaccine programs are now being developed and evaluated with a deeper understanding of the economic and social implications, which results in higher benefits in society and wider adoptions. Vaccinations also now aid us in the reduction of our healthcare burdens and costs, makes it easier for us to travel to different parts of the world, empower women socially and economically, promote economic growth, and enhance and promote equality and equity. I don't think I can imagine the world without vaccines. Vaccinations and other public health measures such as sanitation, clean water, and antibiotics provide significant social and economic advantages. It has been suggested that the economic impact of vaccinations should be assessed in a broader sense than just averted healthcare costs from prevented illness episodes.
that's all for our episode today about vaccines. Thank you for watching.